So two things we are going to do. NCBI we are going, and we are going to Swiss model at the same time. So first thing is that you get a protein sequence. I got a protein sequence, which I have put it in a file. I only know the name, so nobody knows. I mean, I know because I collected it, and I don't know. So I say just say sequence blast this sequence, and I'm copying it, and I can give you in the chat also. So you can also practice if you have. Where is the chat option? Okay, yeah, chat option is there. Chat each meeting is only available to, okay, so team members have the chat option. I don't have the chat options. So we will see them in the sequence, yeah, or at the end of the class we'll get it. So sequence will be available, and sequence will be very easily available to you shortly. Just a minute. So I'm going to NCBI, and I'm saying this is NCBI, let it open. And at the same time I'm going, so this is Swiss modeler for modeling. As I told you that coronavirus models are already available here. Okay. Even for UK variant, yeah. So all the databases. So what we need to do is that I'm just giving the sequence.
Sir, if you need, you can send the sequence to me, like email, and I will put it on chat box if required.
Okay, I'm sending you.
So that people, students can practice.
Yeah, so I'm just sending you. So let me have, my name is not important. So you are seeing the mail is going to you. Okay. So sequence you can give it to student. Okay. So what I did was I just added the sequence, and the program, the Swiss model is working from 1995. So they have optimized in a way. They have picked up already a target by adding me a sequence, and I can say search for template, build model. I just directly say build model. I have not that much time, or I'm doing for the first time. I just want to play with it. I just said build model, and it will build model after some time. But I still don't know what is it. So this is the NCBI. As I told you, there's a very big resource, and all databases are here. Then you have a lot of databases there. Some of them will homology modeling, literature, genome, genetics, protein, domain, structure, taxonomy, training, tutorial, variation. This side you have PubMed. Most of you, if you have worked in any biological or biotechnological project, PubMed will be literature mining. Bookcell will give you a book, knowledge of book, freely available book they have. Blast, then again, a lot of others. So we are going to have a Blast, as we promised that we'll use the Blast. So if you are from computer science, Blast is a web-based tool, plus it is also available for the downloading locally and putting in your own server. Own server on own laptop, and you can download it and use it as a stand-on software. So assuming that I am talking to more people from, some people are at least from the IT background. So we will have some look. So now you see that this is a Blast algorithm, which is basic local alignment search tool. What Blast is, if you are blasting your nucleotide sequence to Blast, you will do nucleotide. If you are doing translated nucleotide to protein, you will say Blast X. If you are taking protein to Blast on a translated nucleotide, you will say this. And I am doing protein Blast, so I am taking this. Okay, so I'm just taking this. We don't know the name of the sequence, if you remember what I have sent you. So Blast will first of all give the name and also the alignment, what kind of species, which taxonomic group of animals it belongs. Okay, now I'm giving it. So I added the sequence. So you see the Blast job title itself is taken the ID I have given. So that is the advantage of the program is knowing input will be Blast file with first line is a just identifier. And then you have different databases. We are just keeping the default database. There's a lot of databases and all. And of course, you can go and read here what is a database. Non-redundant database means there will be no repetition of the same sequence. And blast and in a blast protein blast itself, there are several other blasts. I'm just taking the standard one. And then you can also change the algorithm parameter. I'm not changing anything as of now. And let's run it, and then we'll see what's happening. And here this guy is already making a model. It's still running. Yeah, auto model is made. And it is taking time. So it has to build a model. So it is taking some time, definitely. So here as I ran, and it ran now, and it's running. So if you have a question, you can ask without trying out for anything. And let me drink some water.
So participants, you can mute yourself and then you can ask questions, or you can type in chat box so that I can communicate with them.
If you have a question, please ask. And yeah, so RTC here is the model already built up. We'll go to it, and let me see if the BLAST has given result before and then the model. BLAST is still working. We don't know what was the protein that which were blasted, but what we see here, the model is blasted to a very low sequence identity template, and this template ID is ID of their structure, which is already known in protein data bank, and we can go to click it here. So this is ID is given. So based on this model, we can go from here. So anybody doing crystallography will put it in PDB, the protein data bank. They have to put the structure there. So it's modeling. Our models are built on those as a template. So there shows that it is 12.6% identical. It has a coverage. In the beginning, there's no coverage, and then there is a coverage. And then what is the name of this? So there's some peptides like protein from some which species is still not clear to me. And then it is computing different statistical parameter, making comparison. In model 2, there is no comparison. In model 1, there is a comparison, and you see which region to which region are modeling. So as it says that this region will not model, here in the blue region is model. So the initial something is not, initial 60, 70 are not model. And then there is a modeling happening, and then at the end also something is not model, as it says. Then this is also missing. So this is how what this model will model. Then you can visualize it here. You can even download it. You can take picture of it. They can change to different models here itself. Ball and stick model if you want. So different models are possible, and the blue regions are favorable and red ones are not very favorable here. So only few residues are favorable here. This is logical because this is very low sequence identity, as you see. Okay, so but we still don't know from where did I got this sequence. So let's go back to the BLAST and see that. So BLAST has result, and now it says BAM 46 from Drosophila melanogaster. So that was actually I has taken. And so in BLAST, it will hit to, if you give a sequence, it will hit to the known sequences in database. I have taken it from NCBI itself. So therefore, the first hit is identical, 100% identity we saw. First hit and maybe even more hit actually. Yeah, so the first hit has the fruit fly name, and it is this sequence. And then I can look in a graphical summary here. They will have a, okay, so this says please select the sequence. Okay. Okay, so then this is showing you that this is the
Hello, sir. Excuse me.
yeah, yeah.
Actually, your explanation is not matching with the slides.
What is not
The
matching with the
slides are
slide?
running behind your voice. Yeah, the slides are
Okay,
little
okay,
late.
okay. Let's talk slow
And
then. Yeah.
can you please use the cursor, please?
Okay, sure. अब कर्सर कहाँ से आएगा फिर stop के अंदर? Uh, do we have a cursor in the if the cursor is available here? No, the cursor is okay. So I'll talk slow, yeah. So because the cursor on the web browser, I don't know whether there is a default cluster cursor available. Okay, so let me talk slow. So what I'm going to do is that what I did was okay. So you have the blast, and then what we see here. Maybe I just zoom in. Okay, so what we see here is that the sequence is BAM46 from Drosophila melanogaster, which is first hit, and that was indeed the hit actually. And this is we can have different ways one can look into this. One can look into the alignment. So I'm clicking alignment. Okay. So this is shows an alignment. Query is the hour sequence, and subject is the blast searched in the blast since identical sequence, so it is 100% identical. Right. So this is the sequence 338 amino acid long, 100% identical here it shows, and then 100% positive. Identity and positive identity means indeed identical residues, totally identical residues, but positive means similar residues when there is a change. So all identical is positive also. So therefore, it is 100% identical. Positive will be also 100%. And if there is a gap, that these values will go down, and maybe we can have a look into another one where we'll see the gaps. And then since this one has no gap, so we are seeing no gap percentage. And then the gap, if there is a gap, so this will positive will be less than 100, identity will be also less than 100. So let me go back to description. I'm going back to description again. First few are 100% identical, so we have to leave them out. Okay, so this one is 68%. So let me go there. A distant, yeah. So then what we see here that this is okay. So this will be still not coming. Description. So I'm going to go down where the identity will go down. Okay, maybe this one, last one I'm taking. Hope it comes. Not pairwise. Okay, we have selected all those three sequences. Okay, description. This one has to be removed. Okay. Okay, now I'm clicking. Okay, so here you see that it's not query versus subject is not 100% identical. Positive ones are positive. So L is also a kind of positive, which is computed here. So 52% is identity. Plus they have counted all the position which are positively assigned. And therefore this positive is higher than identity. So positive will be always higher than higher or at least equal. In case of 100%, it will be equal to identity. And there's only one gap, still 1%, still 0% because only one place they are saying gap. So gap is here where the query has no sequence in the subject. Yeah, so that's what we saw. And then what we are going to look, so in from this, suppose you go back to here to description, one can select maybe first, maybe select all the sequence. One way of doing phylogenetic analysis is that in the BLAST algorithm itself, one can do the phylogenetic analysis. So you have to just click here. You can do first round of phylogenetic analysis here itself. So it is showing you the all possible different type of sequences with respect to whichever sequences. So it was Drosophila, so it is matching to beetles or anopheles, so closely related species. And it is making blast to the making a phylogenetic tree to the.
Sir, we are not able to see anything. Hello, sir.
Yeah.
We are seeing only NCBI page.
Yeah, I'm on an NCBI page only.
Yeah, right.
Okay. So I'm saying that the phylogenetic tree can also run on NCBI itself. So depending on the purpose, and that is publishable also. Recently, we have published the same, taken data from here and published actually. So depending on what you are how we are telling the story. So and this is one way of doing. Suppose you want to see the sequence alignment, that is also possible. You can all go to multiple sequence alignment, and then unfortunately it will run here only, but you will see a cobalt based alignment of the sequences here. So I've taken all the sequence versus all the sequence. Alternative to it is that you download selected sequence and do the multiple sequence analysis and do the alignment. So let's see, you will see alignment here. Okay, so these are all the sequences. Now you will see at the bottom, you will see the alignment. Okay, and then it's a compact alignment. You can go expanded alignment. Yeah, you see the full detail of alignment, and then there are sequences which have no region here, so they are gap, and it's a very big alignment because we have used all the sequences which were hit in the blast, and there are conserved region you see, and then there will be non-conserved region also. So that one where there is a gap, here there is a gap. This sequence has no residue conserved at this position. So these are these are because of mutation or error. So yeah, so you see that most of the sequences do not have the sequence at this position, but then maybe there are some which we have there, and therefore yeah. So these are some which have it. So this the gap. Only few of them have, and only one has the complete sub-region. But when you are aligning with those many sequences, you will see gap because of this region. Okay, to make it even nicer for you, and also I'm just taking first 5 sequences, and then one can say GenPact, or I can say download complete sequence. Okay, so I'm just downloading the sequence. Or I can say GenPact. So this will bring you back to NCBI. Sometimes this makes an error. Mm-hmm. Normally it should download, but it has some issue in downloading here. Select new column. Okay. Why not downloading? Okay, so if it is not downloading, still we can download it from clicking to the access and IDs. So yeah, so I went here, I have the first sequence. So I have taken the first sequence, which is same as, I've just copied it, which is same as one which we have it in the alignment here. Sorry. Oh, it's copying only to the top. Copy here, please. Oh, not here. Okay, it's copying. Okay, so this is first one, so identical one. So if we want to make an alignment, we can use online tool like Muscle. If you want to make an independent alignment, maybe I just take 3 sequences or 4 sequences and do the alignment in Muscle. Muscle aligner. Alignment. So, yes, muscle alignment is available from NCBI. If you are using a local computer, one can download it in Linux and use it. Also in Windows with terminal possible nowadays. And so I have one sequence. I just want to take few more sequences. And also I just need only this click here. Okay. So okay, so I have taken this sequence. Suppose I just want to align both of this, yeah. So you just make an alignment. If you have 5 sequence, you can take 5 sequence. If you have 25 sequence, you can take 25 sequence. Okay, so you'll get an alignment. So you're seeing the alignment. So where there is a, I have to zoom it again, I guess. That's better, yeah. So you're seeing all the stars means identical positions and similar is double dot. So S is changing into A is double dot. And it was close sequences, so you expected them to be nearly identical. And so that's what it is. So this is how the alignment is. And now suppose I just want to make this line as a sequence logo, or maybe just half of it, but let me, so I need to fully take it on. So if I want to create a sequence logo out of it, web logo. So I have to go to web logo and web logo 3 from Berkeley. Yeah. This is possible to create it here. So for web logo, you don't need ID, you just need the sequence. So now I can decide how big I want to make. Maybe first half we don't need because it will be too big otherwise. So from TP, because we wanted to have the one changed position, and let's run it. So by default, you can run it and you can change color and also. So I'm telling it is amino acid, or it can also detect automatically, or it's a DNA, RNA, whatever you want to define. So it will detect automatically. Okay, so it's not giving anything. So let me go back here and ask what is the region. So let's see. 1 to 34. Maybe the base is too big. Okay. Mm-hmm. There might be a bug in the web logo 3. Yeah. Web logo 3. Okay. Maybe I just give full length and see what happens. Okay, it created actually, and it created for most of them. Most of the positions are with very big, and only one position has a change. So that was quite obvious. And then in this one can take it to the wherever you want to present. And let me take you to the example which I have taken in the figure, which you normally take it. So we just take this. You can play with your own name also, create a logo. It's very often used in the, yeah, so this is T and F are two residues possible, and rest of them are conserved. And bioinformatics, because a big letter, bio, translational bio. Here it has changed. Bio, it has transfractional. Maybe just remove few of them, then check. And then you can also change the logo size large, sequence bit. You can make other output high resolution, low resolution, maybe better. Okay, so now this one is large. Yeah, so this one has this is how you will create a logo, and somehow the web logo from Berkeley was not working. So let's see again. Well, normally it works. So let me check again. Okay, so it's working. So you see that there it was. Yeah. So T and F is changing in first position, and then B and M at 14th position is changing. So wherever there is a change, there will be multiple residues, and then it is also telling you the size of O is telling you how many times it is O at this position. So single one is bigger than length, and one which are half of or less than half of their size is also telling the number of times the residues is repeated or alphabet is repeated. Yes, so this gives us a whole idea of how web sequence logo is done. You have the sequences here, and you want to make it do some more analysis, right? So one of the analysis which we often do it is that you saw the domain. So you do go to the HMMR, which is hmmr.org, and then you want to have search. You want to search what kind of protein domain this sequence has. So we have a sequence. You saw multiple sequence. We have a sequence which we have used in. Okay, we have the sequence. Here we have the sequence. I don't know what sequence it is, but one can get what kind of protein domain it has. So I don't think it takes this line here. So I'm just removing it and trying to run it. So it will show me the whole domain and also do HMM based. Oh yeah, so this is saying this is a hydrolase domain, serine hydrolase domain FSH. Okay, so this is the position of serine aminopeptidase is shown, and then there are two different domains are there, and there are some signal peptide and transmembrane domains, but these are main domains, and this is potentially a reactive site, predictive active site, serine 187. So you will get it, and this is again done by blast-like analysis, which is HMMR will search for the sequence, and then sequences are given probability in the now. And it is also searching first with the Drosophila because it was Drosophila sequence. And so look for all Drosophila sequence and other insect sequence. So this is HMMR. So basically, if I have to draw it, I will just take this information, each piece of information, and then draw it myself, a nice figure for protein domain. And when we are doing homology modeling also, we saw that this sequence and this sequence are not matching, right? Half of the portion is matching and half is not matching. This is because this protein domain here plays critical role. So the domain are known. So we saw just now that in the beginning, 105, and at the end, 30, 40, 40 residues are not mapped on domain. So if domains are not known, then a structural component elements are also likely to be missing, and that's what the iTesser is doing. But no, sorry, Swiss model is doing. But alternative is to use iTesser. And let me see if iTesser is logged in here for me. So yes, sir, I'm calling. But iTesser do take care that it will map all the reasons, irrespective of. So it will overcome the problem Swiss model is facing here about modeling left and right. It will model until here using this template, and then it will use another template to model the missing portion. And that's how it will fulfill. So this is much advanced. And again, in this one also, COVID-19 is already provided. So if you want all genome-based sequences of COVID-19 is available here. Models are also available here. So if you are interested in docking something, you just have to take these models. So this all are available here. And so that it save time because they also know that a lot of people will be using COVID-19 data set for pulling into homology modeling. Why not put once in a while we put it, because then they will save a lot of server time. So this is going into server, and then we need a iTesser. We need to pull our sequence here, and then we need to give an official email ID. If you have it, you can give it, or you have to first time create it. So I'm just giving my ID, and I'm building a model. Let me see if my email ID has the model. There has the password here. Let me have a password in a minute. so ITSR password. But ITSR takes some time, like it run ITSR. Okay, so ITSR takes time to run, at least one day it will run, so not less than that. Let me have a password for ITSR actually. I'm just looking for it. So there are two restrictions of ITSR, that one you can only run one analysis at a time, and then the second problem is with ITSR is that it will run 1 to 3 4 days. Obviously it will be full model, so convincing that model is better is easy. So let's see if mine is running. So this says sequence is submitted. Okay, so this somehow it has forgotten my my email itself because I have not used it for several months. Maybe I made it name Matix. Okay, Matix. This was error. So I have to get the password again. Some code it will give actually. So if you are looking to use it, you have to be ready. Create a login password. SQ5. Okay, so this is running, and this is going to run for some time. Okay, so just I have to keep this. I have to track this. It will take some time. So if I'm tracking this, I'm getting that, and they will keep this. They will keep the data on server for some days. 60 days they are saying now. Depending on how much server load they have, they will change this date. So you will get another way of doing the analysis. Another way of doing the analysis. There are several ways you can also create a phylogenetic tree alignment. One of the quick phylogenetic tree making software which is possible to teach in the course like this phylogenic.fr. Phylogeny.fr is a website where we are phylogeny.fr. Phylogeny. So it's a French website. We'll definitely get it. So there one can load a small. Yeah, this is there, right? So here one can load a small analysis. Suppose you are looking for 10 sequences of phylogenetic analysis. You can load a sequence. Maybe I have a sequence here. I'm looking for a faster sequence. MTV fast. And let me have a look in my Notepad++. So Notepad++ is something which you will need to keep to install, because that's easy advanced version of Notepad. So it will keep your file stored if you have one running. So let me see. So we run a lot of different type of files. So getting a sequence file, I just wanted to use a sequence file maybe on desktop. Okay. Just trying to see if we can get a sequence file. If not, we will use one from somewhere. Just a minute. So for the alignment purpose, where again we get a good sequence. We need 10 sequences to run this analysis automatically. Okay, so let us generate it from the NCBI. So this NCBI was, these 5 sequences I've selected, but I'm trying to download it. It should download it actually. Normally it automatically downloads it. If not, we will use, okay, in between we are going to use NCBI again. So I went here, and then in the tutorial I have said that we will use Serpin A1. Serpin P has to be there. Serpin PIN1, and I'm looking for it. So then what happens that Serpin A1 is available, and from this, by doing this, you will go to different databases and different tools. We require to go into protein database. So we wanted to download some protein out of it. So we started getting sequences. So these are the sequences. So I'm just selecting this sequence. We do not want to say partial sequences. 400 something has to be the length. These are partial sequences, and synthetic one we are avoiding. And one per species, one human maybe, yes. We do not want to repeat one from rat, one from this, one each from different species. Okay, one each, so that you get a kind of good distribution. So I'm randomly taking some sequences, one each from different species, and yeah, without looking into much detail. Now this can be downloaded at faster text. Yeah, so easily from NCBI one can download, as you see. And now this is kind of a sequence which we have. So when you are seeing a FASTA as an example, you see that how long the code can be, starting with the greater than sign. So this goes for several lines, but they are all together in a single straight line. So therefore, it's not a problem to have it. And then I will show you. I just take this to my Notepad++, and Notepad++ will keep track of. So you see that this single large line. So there's no problem to have it. And then one can keep it if you want. For simplicity, I'm reducing it, because in the alignment anyhow it will not be taken. So, but you no need to do it. So let me take this whole sequence and check in the phylogeny.fr if we get a way. So phylogeny.fr is not. So it says robust phylogenetic analysis for non-specialist. Okay, let me have a look. It says one click. I'm going for one click. It says add your sequence. Okay, I'm adding the sequence. One can also choose from the other way. You can save the file and run it. So I've just given the sequence. I'm just say course practice. Okay, and then it will ask some email ID. Okay. And then I'm giving it email ID. Matrix. Okay. Is processing. Another way of doing same thing is that you do the alignment it yourself one by step by step. You do the alignment, then it will align. Something is aligned here. Now it's get aligned in. I'm doing it in muscle. Okay, so you do the alignment, and it will take some time, and this is running automatically. So depending on how much resources, there are several ways of doing the same analysis. These are browsers, so basically anybody can download 10 sequences from NCBI and do it. So another thing which I wanted to tell you is that tomorrow also we will be covering. Ensemble is also a genomic resource database. So if you look for any gene in Ensemble, says SERPIN A1, you will get an SERPIN name is correct. You should get it. SERPIN A1. Retry in few server failed to respond. Retry in few seconds. So there must be some problem. Something is wrong. So we'll try tomorrow. So, but it normally works. So, okay, maybe the Asia one has problem. Okay, let's see. Ensemble Asia. Okay, maybe just taking the gene they have. No, this is also not working. So there's some problem in the Ensemble today. So we'll try it tomorrow, and you will retrieve a lot of information from there also. And that's what we wanted to tell you. And then this is while this is running, we can go. So this is you see this is alignment. Okay. So they start varying the position because they have taken randomly from different species. This is fine, but what you see here is that some number and all. Right. I can use it now, but when you're making phylogenetic analysis, you take the sequence, you'll start checking what can be the problem. If this is big one, you chunk of this, you need to rename the sequence actually, so that when you are doing the alignment or phylogenetic analysis, you see the meaningful outputs. So here this again you have to search because 35, 53, ending with 99, 39, 28, we don't know by name which species which sequence it is. Only for like say alpha 1 antitrypsin underscore human, it is visible. Only one of them. So it is advised that we clean the sequence and rename the sequence when you're doing phylogenetic analysis so that there is a meaning. Since this is a practice and this is a small number of sequence, we can still manage. Right, so let me download this sequence and we will have a run on local machine, the phylogenetic tree. Okay, so I have the sequence and we see that muscle alignment is here. And what we need to save it as we are going. So what we are going to have put it on desktop for time being. Say sequence alpha 180 ALN, and ALN is there for alignment. So I'm just doing this. Okay, so, and this is random alignment. And now what we need to do is to find that tool called muscle, yeah, sorry, mega. Muscle, we have already done it. Okay, so let run, let alignment be. So I want to build alignment from edit or build the alignment. Okay, let me open like this first. Okay, so I just missed something. I double click it actually. So it was asking a question. Okay, so align, build. So it is asking retrieve from file. Okay, I'm saying retrieve from file. And then I know that it is on desktop. So it says fast. All I am saying is that this one. Okay, so if you had taken the sequence, raw sequence, you could align again. There's a muscle and alignment, but since I have already aligned, I want to save the session. Export alignment session into Mega, and then MG. Again, I go to desktop and save it. You can name it whatever you want to. So I just say alpha 1 AP, or you can leave it blank also. So now it become make file. The make file is required for building phylogenetic analysis. So I'm going here and I'm saying there are different phylogenetic tree building method, neighbor joining method, maximum tree, and maximum likelihood. So we'll just make one of the tree, and make file is required. So it's asking for 1000 bootstrap. I'm saying yes, just running it, and you will see the tree. So this is the fastest method. So you see the tree, original tree, bootstrap means. So these are different tree. Now in this one can change the shape. As I told you that we will practice that circular tree. So this is circular tree. We can make it down. Okay, and then it will become, you can radiate the tree. You can make different ways of tree. You have seen, but still, I would say that still the names are not clear. Since it's a small file, it's okay. You can go back and trace here also. You can edit it actually, rename it by cutting pasting from somewhere. So it's possible to edit it. Suppose I just want to place this name by something like that. It's possible, but if it was 1000 sequences, yeah, how many time you'll edit if you have to edit? So it's better to restructure enough beforehand, rename the sequences and go for it. And these values are bootstrap values. Bootstrap values are statistical significant. It is telling that this subtree, which is flowing from here to here, and this one is sub-branching, and these are routing. This sub-branch is 100% time here only. Out of 1000 trees, they randomly made, they found that 100 times this is there. And then this is 79% means these two are at this diverging from this point is 79 times 79% of time seen when you make 1000 random trees. And this software Mega is also providing an initial hint of the what analysis you have performed, so that it's also created you a legend of the figure. It is not necessary that it is giving you correct one, because you need to know this is Serpin which we have used. Evolutionary relationship of taxa it is written. Instead it is evolutionary relationship of alpha antitrypsin from selected species, and then you have to name the species, and then you obviously, but you get a good hint when you are making the tree. And also you can make a colorful, so you can change the way the branches are looking. Suppose you want to make line by 4, and some other changes. So you just need to play with it actually. So the bar has changed. If you want to change the color of the bar, color of the label, font to maybe red. Yeah, so basically you can play with different things, and then you can copy. If I have copied it, I can have copy in my PPT. Why you can get copy, you can save it. So there are different things we can do in Mega X, and then there are different algorithms, and different algorithms are used for phylogenetic tree, which you use. This is the fastest one, which runs faster. If you have even 1,000 sequence, you'll get result in the same day. Other methods are used for specialized purposes. You'll get result, maybe a better result, but there's no hard and fast relationship that you will always get a good relationship. One thing we have to remember, to clean the sequences, not only in the terms of name. Suppose there is a sequence which you see only partial. I have deliberately taken full length sequences. Those sequences will create a problem because 400 positions where the information is stored, if you have only 150 points to in one sequence and remaining is blank, it will create a problem. You will not get usefulness. So those things we have to replace. And then you ask then what should I replace with? Suppose you have a sequence of mouse missing, is partial. You will use a sequence of rat, a relative species, closely related species, rather than partial sequences. You just need representative sequences. You don't need all the sequences from all closely related species. From rodent, you need one. From bird, you need one or two, or whatever number you want to give, maybe five. But then if it is partial, or a lot of times these alphabets are XXX, then those will not work actually. So like sometime you see like this. So this is, again, you will not get any information out there, because in the sequencing technology there was an error which led into problem. And so each point is providing an information which has to be stored for making the phylogenetic tree. And the tree will be very different. We have 27 sequence or 25 sequence, or correct sequence or wrong sequence. Based on that, the tree will become different. What I mean by saying that, the sub branch will go here and there, and also the score will go here and there. So this one has to take care when you are running. Okay, so and then if you have a very big tree here, in Mega and other tools also, let me make it linear. So we are seeing like this. Suppose we just want to not see after this point. So here there is option to, we can name something, and then if I name it X, suppose it will be visible. Yeah, so this will, I just deblock it. I mean, I just compress it. So this there is no further sub information coming out, only one single information coming out, that X is the information, and so the value inside the X is not very precise. If you don't need it, you just can say. If you need it, you just can open, and then you are saying that these are X. Okay, so you can name draw it differently, and the more you practice, so you try to get some more meaningful sequences. And every time, suppose even if I have 550 sequence in one stretch, I'm not getting the final result. I need to modulate here at least 10 times before I decide, okay, I'm going for this tree. And this is the my method of choice. So this is hope this is giving you a good idea of using how to draw the phylogenetic tree. And this, while this is happening, let's go back to the slide, to the PowerPoint where we had. So we had built up this we already built up, so we don't need this. We don't need this. Will run and give you the result later. And this one is still running. So this is bookmark the website, so it's still running. So you see that the server, yeah. So this has already run. In this one, it is giving you an idea, yeah. So if I can zoom, or maybe I just open as a PDF, we get an. So this one is taking care of this name, so that you know the camel. I think this shape. This I don't know. So you can have, or we can always go and have a look, because remembering a species name, I'm not giving IS exam here. So obviously this is a kind of a baboon. So I don't normally try to remember this species name. And then what is this rhino? Let's have that. Yeah, so this is also kind of a monkey. So the monkeys are coming together. And these are primates. So when we have knowledge, it is all primates are going together. Then this is good. Let's see, what is that? Camel. Yeah, this is camel. And this is, I think it's ship. Yeah, so this is a this is ship, right? So ship and camel going together. Rodents are here, close to human, but primates are going together. So now one can easily explain what are the subtrees. And then this legs, and that fish will definitely go away, far away. So that's what it is, because all rest are mammals. And then let's see what his leaks is. So just a minute. हाँ, हेलो सर, मैं जरा क्लास में हूँ, जरा कॉल करता हूँ मैं आपको क्लास के बाद, हैं. So leaks is you see that it is medium size belonging to cats, so they are coming even separate groups. So if you add more cats, they all should go in this line, and then so therefore the having a meaning is very important. And if you add more fishes, that will go with Danio. And if you add more primate, they'll be clubbed together. So if your objective it is clubbing different mammals together and the subtype of mammals, so you need to add more sequences of that. Suppose you are working with plant, you do not take this species. You need to take Brassicaceae or other species. So depending on what sequence you'll provide, you will get the result. So this is faster way. And here also you can edit it actually. Colorize, outgroup, regroup. So there are a lot of things you can do. Circularize. So I just circularize, it will load. Yeah, so this is kind of circularized, but then you can't read properly. The radial, but this is also a very fast way of making the tree. Okay. So that's fine, right? And hope that you are enjoying. We will have another class on comparative genomics where we'll cover part of this and some new concepts. And if you have a question, now we can answer those. I can answer some of your questions. Yeah.
Thank you very much, sir. So I don't know about the participants, but I really enjoyed it. And now it is open to participants. If you would like to ask any questions. Sir, there was some question in the chat box. So, do you have any opportunities for internship of some students?
Yes, we have a lot of internship going on with the students. You just write me an email what you want to do, and we have. So currently we have an IT student from Jaipur are doing, and at the moment 12 students are doing. So we can design a project and you can work. So that's not a problem.
Okay, okay.
And we will have a week. You will participate in our weekly meeting discussion. Suppose you are doing for, so depending on how much time you want to spend, you just write down what your time. They said you want to do it for 3 months, is a master thesis, bachelor thesis, or just we want to learn. So there are different kind of internship possible.
Any other questions? Please unmute yourself and you can ask.
So if not, so you can ask it tomorrow also. So today's
Yeah. So because sir is also available tomorrow and on 22nd also. So we have sufficient time. So for the time being, sir, we should end up with the session. Thank you very much once again for the informative and some practical implication and a lot of resources that you have shown to us. So definitely that will be useful, very useful to all the participants. So we are ending the session over here. Thank you, sir. So we will meet tomorrow at, sir, your session is going to start at 3:15, I feel, 3:15 p.m.
Yeah. So
Yeah. Okay, sir.
Yeah. So thank you. So yeah. So thank you. First, thanks for everything, and let's meet tomorrow again. Yeah. So thank you. Dhanyawad.
Okay, sir. Thank you.
Okay.
तो मैंने end कर दिया।